What is a drug? Now this can mean different things in different social contexts. So for today, we will think of this as a compound or product that cures or alleviates a disease. I was going to start with sort of a disease or some ailment in patients. First, this needs to be recognized, identified, before we can make any progress. And then to come up with this ideal drug, we're going to need to go through a few phases of research. And the first one we'll call preclinical research. So this is done by scientists at research laboratories, at research universities like Harvard, Tufts, BU, all across the country and the world. And the goal here is to understand how the disease works and try to come up with a potential drug that could affect that disease state. The next stage is what we'll call clinical research, and this involves testing in humans, testing that drug in humans. And previously we had these scientists and sort of research institutions involved. Now there's a couple more key players. The first one is what's called here a drug sponsor, or we could think of as a pharmaceutical company or biotech company. Somebody with a lot of money, because clinical trials are very expensive. So you really need a group like this involved to sort of push the drug through the next phase of testing. But they are going to be working actually very closely with the Food and Drug Administration, the FDA, which is the regulatory body in the US. It tends to be a very lengthy process. On average, it takes around 14 years to go from sort of identifying like what it is you want a drug to do, identifying a target, to actually getting FDA approval. Of the drugs that actually enter clinical testing, enter testing in humans, around 85% of them don't get approved. We come up with a really big number for, on average, then how much it costs to come up with one successful drug. These failures happen at each sort of stage of this process. So in the evaluation of safety, around 33% of the drugs that enter phase 1 trials don't make it past. They're deemed not safe or not safe enough to make it to the next stage. And even of those drugs that are found to be safe and go on to be tested for how well they work, 42% of those are found to be not effective, or again, not effective enough to warrant going on to the next stage. And even those ones that did show a pretty good effect and they went on to phase 3, well, around 60% of those are suspended as well. Research models of disease, laboratory animals, test tubes are not the same as humans, as much as we might like it. There's just biology that's different. And if we did try to study human biology more specifically, unfortunately, the samples or materials available to us are pretty limited. Each new drug that goes through this process needs to be somehow better than a current drug. Now that a number of drugs have been through this process, they're found to be safe, they're used for particular treatment of particular disease, now they're actually available for scientists and doctors to think, to try to apply them to another disease. Another way to try to address these challenges is to improve our laboratory models of disease. So one way to do this would be to sort of better understand the current models we have, know what their limitations are, know that they can't really predict how everything is going to work in a human, and to really try to come up with new models that can provide better examples of humans. And the third thing is to come up with new technology that lets us do more with less. And this really gets at the opportunity to try to learn from human samples. And one particular example, which I think is really exciting to improve this process, is this thing called a tissue chip, which looks like this. It's a piece of plastic around the size of like a quarter or a key. And the idea is that you can put some human cells in little like wells here, little areas, grow these human cells to mimic a human tissue. Before you actually go to testing in humans, use this model that uses just a little bit of human cells and evaluate toxicity of drugs at this phase. So MS was first identified as a disease in 1868, kind of a long time ago. And it wasn't until around 100 years later, in the 1960s, that the first animal model of MS was developed. And this was done in rats and in mice, and is still used today. So here's kind of what they were thinking. They thought, how do immune cells enter this inflamed brain tissue? Maybe there's an interaction between some unknown, so far unknown molecule on an immune cell that's going to interact with a receptor on a blood vessel that goes through the brain, and then this interaction will allow a cell to enter and cause damage. But if they could come up with some sort of compound, some sort of drug that would block this interaction, then the cell could go by, it never enters the brain, never causes damage. So to approach this question, they considered all these receptors that had been identified by other scientists and said, maybe one of them is important in this case. But there was one that they identified that was critical for these mice to get the MS disease. And they partnered next with a pharmaceutical company to alter the drug a little bit. Previously it worked in mice. They needed to make a version that would work in humans. And then started to take this into the clinical trial phase. So we know that natalizumab prevents immune cells from getting into the brain, which is really great for treating MS, right? Because those immune cells are the ones causing damage. But in the case of a person who has the JC virus, if this virus gets into the brain, in a healthy person, immune cells can go in the brain and take care of that virus, keep it from spreading too much. In a person taking natalizumab, now those immune cells can't get in. Now that virus can sort of expand out of control and lead to extensive damage in the brain, which, as we saw, turns out to be fatal. And the FDA felt that because they could identify the JC virus as this key risk factor, they felt comfortable reapproving it.